First, let's talk about drought-resistant and climate-adaptive crops, as extreme weather conditions threaten crop yields. Scientists have developed new breeds that can thrive in harsh environments through advanced biotechnology, including CRISPR gene editing and selective breeding. Crops such as drought-tolerant maize and salt-resistant rice are becoming lifelines for farmers in water-scarce regions. Artificial intelligence is transforming modern agriculture. AI-driven weather prediction systems analyze vast amounts of data, from satellite imagery to historical climate patterns, offering farmers accurate forecasts.
Molecular markers are essentially biological landmarks or tags found within organisms' DNA that help scientists identify specific genes or traits. Just as a signpost on a road tells you how close you are to a city, a molecular marker tells researchers they are near a particular gene, such as one for disease resistance in crops or a specific health trait in animals, because these markers are often inherited alongside the gene of interest. Genomic tools are the advanced technologies and methods used to detect these markers and analyze the entire genetic blueprint, the genome. DNA sequencing and PCR polymerase chain reaction allow scientists to read the DNA quickly and accurate. Together, markers and genomic tools make it possible to breed crops and diagnose genetic diseases.
Next, the molecular foundations in plant conservation. First one is applied DNA-based tools to assess plant genetic diversity. Molecular markers and genomic data are used to measure genetic variation within and between plant populations, which is essential for maintaining species resilience and long-term survival. Second one is identify priority populations for conservation. Genetic analyses help identify populations with high genetic diversity or unique alleles that should be prioritized for protection and management. Third, detect genetic erosion and inbreeding risk. DNA-based studies reveal loss of genetic variation and increased relatedness in small or isolated populations, allowing early intervention to prevent population decline. Last one, support restoration and conservation planning. Genetic information guides the selection of suitable source populations for reforestation, habitat restoration, and reintroduction programs, ensuring genetic compatibility and long-term sustainability.
Next is about genomic tool and the application in plant conservation. There are two tools that we use in this application. First tool is environmental DNA, which is eDNA. eDNA is a genomic approach that uses a plant species identification technique via an air or soil sample. The application is biomonitoring endangered species without requiring physical sampling of the plant. Two advantages by using this tool. First is non-destructive. We need to uproot the plant, and this can keep sensitive for the habitat instead. Second is efficient. One environmental sample can detect hundreds of species at once. The second tool is whole genome sequencing, which is WGS, a complete genomic tool that maps all the genetic instructions of the organism. The application is for identifying risk causing mutation and determining the adaptability of plant to climate change. The advantages by using this tool is we can get more detailed data.
For my part, I will present a case study on Vietnam Mokasa Dawe, a rare and endemic aroid species discovered in central Vietnam. This species occur in small and fragmented population and shows strong morphological similarity to species in genus Alocasia, making the identification based on morphology alone unreliable. To resolve this problem, researchers apply molecular markers and genomic tools. They sequence several chloroplast DNA regions including tRNL2F, RPL20 to RPS12, and tRNK or METK, as well one nuclear gene, phytochrome C. This DNA sequence will analyze using maximum likelihood and Bayesian phylogenetic methods to determine the evolutionary position of the species. Next, we move to the next one, which is the genetic analysis, show that Vietnam Mocassadae form a distinct and well-supported clade, confirming that it represents a new genus that is closely related to Alocasia. This molecular evidence prevented misclassification and provides strong support to recognize Vietnam Mocasia as a separate taxonomy unit. Based on its restricted distribution, small population size, and habitat distribution, the species was assessed as a danger under the IUCN Red List criteria. This case study clearly demonstrates how molecular markers and genomic tools are essential parts in plant conservation as they enable accurate species identification and support effective conservation planning, and especially for rare and endemic plants.
Next, I will talk about the impact of application of biotechnology tools. First, in the medical and healthcare field, biotech has revolutionized our approach to health, moving from rapid diagnostic like PCR tests to life-saving advancement in mRNA vaccine and personalized gene therapies. Shifting to agriculture or green biotech, these tools are essential for global food security. They allow us to increase crop yields through GMOs, enhance nutrition in food like golden rice, and promote sustainable farming by drastically reducing the need for chemical pesticide. Finally, looking at the environmental and industrial impact, biotech provide vital solutions for a greener planet, such as using microbes for bioremediation to clean oil spills, producing renewable biofuels, and creating biodegradable plastic to replace petroleum-based material.